everyone, welcome back. So you could have seen my previous two-part videos, where the first video I have posted you the introduction of genetics for the principles of inheritance and variation, and in the second video you could have observed the basics that we must know before going for the Mendel's approach, where we saw about Mendel and his experimental methods and what he have done, in which experimental plants he have done all the experiments, and even we saw the 7 pairs of contrasting characters and how to assign the allelic symbols. That is very important whenever we do a cross. Even I told you the mistakes students can commit and how we can overcome that. And after that we are going to an important topic now, Mendel's approach and his experiments. Come on, let us have a look on that. So Mendel's approach. There are 4 major approaches which Gregor John Mendel have done. What is the first approach means? Pure breeding. We know that pure breeding means it is homozygous trait. Pure breeding means what? Homozygous trait. So Mendel took a pure breeding plant such a way that the pure breeding homozygous plant was exhibiting dominant trait and another one was exhibiting recessive trait. That means factors always occur in pair, but here the pair were exhibiting dominant trait, here the pair were exhibiting recessive trait. So the name we can assign for this plant is homozygous dominant, homozygous recessive. After choosing an homozygous plant, that is homozygous dominant plant and homozygous recessive plant, how to identify? He performed a cross called test cross to identify the homozygous trait. After identifying the homozygous dominant and homozygous recessive, we are going to call this P1. He emasculated any one of the plant and artificially hybridized the flowers. And after hybridization, he obtained a plant called F1 generation, first filial generation progenies. Now he cross hybridized the parent 2. First he made P1 to cross, now P to cross. P1 we know it is a homozygous dominant and homozygous recessive. What is the feature of P2 means? It is a hybrid F1 into F1, because you all know that F1 is obtained by crossing a P1 to P1s, therefore F1 is nothing but a hybrid. So here he crossed these two hybrid, he obtained a generation called F2, and only for this you can perform a test cross, Punnett square you can perform for this children. And after this his interpretation was pure and pure mathematical expression. Using mathematical formula he observed F2 generation and he interpreted all his results using the mathematical expression. Now children, we are going to see something about Mendel's monohybrid cross. In the previous board we have seen something about Mendel's approach, and based on his approach only now we are going to perform the Mendel's monohybrid cross. First we should know what is Mendel. Of course you know the name of the scientist, father of classical or father of genetics. Monohybrid cross. Mono means single, hybrid means the plant formed by crossing two different parents. So monohybrid cross, what is defined a monohybrid cross? Monohybrid cross is a cross performed between two different plants. And what is the unique feature I am going to add is differing in one contrasting character. 7 contrasting characters are there. So monohybrid cross is nothing but a cross performed by crossing two plants differing in one contrasting character. Therefore, the type of cross that we are going to discuss today is monohybrid cross. It is a monohybrid cross. I am telling you how to write in the examination. In that way only I am giving you even the notes also. Simultaneously, the way I am going to give you the same way if you write in the examination, there is no chance for reduction of marks in your board exam. Preferably it is a 3 mark. Combining with the test cross, it may come for your 5 mark examination in your board examination. Now here, what is the experimental plant that they have taken for the purpose means the name of the plant is, very good, Pisum sativum, and half mark will be reduced if genus and species are not underlined. The name of the experimental plant is Pisum sativum is the experimental plant. Okay, fine. After this, what we have to go means we have to go for the trait. So there are a lot of traits are there, children. Among them, I can choose any one of the traits, and maybe I can take a seed shape. I will take into consideration. In your textbook, it is with stem height, and perform here with the seed shape is the trait. Among the seed shape, there were two important traits were there. What are the two important traits? One trait is round, and another trait was wrinkled. So one seed was round and other seed was wrinkled, and the round was dominant and the wrinkled one, it is recessive. Recessive trait is suppressed in a hybrid, dominant trait is expressed in a hybrid. That important task, please do understand. Next is, for this, what are the alleles for this? That is very important. The two important alleles that we are going to take into consideration is, based on the dominant trait, capital R, comma, small r. Because RW we cannot put, based on the dominant trait only, we have to assign the symbols I taught you. Now we are going to perform the cross. Now he took the P1. P1 means what? The first parents. So the trait should be pure breeding. So one should be homozygous dominant, because the first is he have chosen only the pure breeding plants. So the parent one will be always homozygous dominant. He crossed the homozygous dominant with the homozygous recessive one. He crossed homozygous dominant with the homozygous recessive one. That means what? Capital R, small r, small r. That means the round seeds was crossed with the wrinkled seed. The round seeds were crossed with the wrinkled seed. After crossing these two, he obtained what? P1 is crossed to form the F1 progeny. The parent one, which is homozygous trait, were crossed to obtain the F1. So what is the F1 progeny means? It will have capital R and small r. How, sir? To understand this, we have to go for the gametic form for this. So when we go for the gametic forms, here the gametes are, as per the second law of genetics, law of segregation, gametes they segregate independently at the time of gamete formation. So capital R, small r and small r. This is the gametes form, which is always haploid. So gametes have a segregated. Now we are going to see the F1 progeny. So F1 progeny was capital R and small r. So capital R and small r, that is nothing but it is heterozygous top. It was heterozygous top. Okay. So second step of Mendel is over. What is the third step? Third step is parent number 2. So what is parent number 2? Parent number 2 is nothing but F1 into F1 is the parent number 2. So we took capital R and small r, capital R and small r. Characters are controlled by a discrete unit called factors. Today we call it as genes. And factors always occur in pair. That is the first law. Third point is, in dissimilar pair, heterozygous, one pair dominates the other, as per the first law of genetics. Second law is law of segregation. Gametes they segregate independently. Therefore, when I am going for the gametic forms, it will be capital R, small r, capital R, small r. So what we have to do after this? After this, we have to perform a small cross that is called as Punnett square. So what is Punnett square states? Okay, I'll take here the Punnett square. For this, I am going to perform a Punnett square or Punnett square. Okay, draw a checkered box, double line for gametes, and inside the double line, the checkered box, what we have, it is for the progenies. So capital R, small r, capital R, small r, capital R, capital R, capital R, small r, capital R, small r, small r, small r. Okay, this is over. After this, what has to be done? After this, what has to be done? I'll tell you in a simple manner. After this, you have to put the first start with genotype. What is genotype? Genotype is the genetic constituent of an organism. That is capital R is to capital R, small r is to small r, small r. How many times capital R is there? Only one time. How many times capital R is there? Two times. How many times small r is there? One time. Okay, fine. Next is, based on this, you have to put the phenotype. You know, phenotype is external observable character. So there are only two possibilities are there. Round is to wrinkled. How many round will be there? Capital R is round. It is a heterozygous, so capital R, small r is also round. Therefore, 3 is to wrinkle is only 1. So this is how till now you have this you have to present. After this only we have to go for the interpretation. So next we are going to focus on the interpretation. Now we are entering into the fourth step of Mendel's approach, that is the interpretation of the results. So 1, 2, 3, 4. 4 interpretation I am going to give, and based on this interpretation, how many laws were framed based on monohybrid cross. Interpretation of monohybrid cross. What did Mendel infer from this observation? The first one is the F1 progeny crossing the P1. We got some F1 progeny, right? The F1 progeny showed any one of the parental trait. That is, in simple way, I can tell you that if it was seed shape capital R, small r, take the gametic form, it was capital R and small r, it was exhibiting round. Round was any one of the parental trait. Therefore, we can understand that the F1 progeny, which is obtained by crossing two pure breeding plants, pure breeding homozygous dominant and homozygous recessive, the F1 progeny resembled any one of the parental trait. Second point, the F2 progeny showed both the parental trait. That's why we got a ratio that capital R is to capital R, small r, small r, small r. We got round is to wrinkled. Wrinkled was not obtained in what we call F1 generation, we obtained in F2 generation. So F2 generation had both the parental trait. That is the second observation. First observation was the F1 progeny showed any one of the parental trait. F2 progeny showed both the parental trait. So here the key word is any one of the parental trait. Both the parental trait appeared in F2. So in need exams, the F1 progeny resembled any one of the parent, law of dominance. Did not resemble both the parents, incomplete dominance. Parent taught you about incomplete dominance till now. The third point is the form of trait that appeared in F1 is called as dominant. And how many times it appeared? It appeared 3 times in F2 generation. That is capital R, capital R, capital R, small r, small r, small r. These two were giving a ratio called 1:2, 2:1, and it was tall ratio, so means round ratio, 3:1. So how many times we got the dominant trait means it appeared 3 times in the F2 generation. Okay. So what is the last inference? The two contrasting traits did not show any blending, that is as per the second law of genetics. So mathematically the equation is, taking the gamete, we have got the ratio as 1:2:1. I don't want to explain this. A plus b the whole square formula is obtained. So what is the law framed only based on monohybrid cross means the first law is called as law of dominance. Characters are controlled by a discrete unit called factors, today we call genes. And how the factors were appearing in the parent, it was not as independent. Both of them combined together occurred in pair. So the first law of dominance is characters are controlled by a discrete unit. It is called factors. How the factors are always occurring? Factors always occur in pair. Homozygous dominant occurring in pair. Heterozygotes occurring in pair. Homozygous recessive occurring in pair. Third point in law of dominance pair means dissimilar pair in heterozygotes. Third point is for heterozygotes. In dissimilar pair, one pair dominates the other. F2 progeny, capital T and small t, or capital R or small r. We can take anything. In dissimilar pair, one pair dominates other. Capital Y, small y. Yellow is dominant over small y. Capital Y is dominant over small y, exhibiting the dominant trait called yellow. So again, I'll tell the three statements. Characters are controlled by a discrete unit called factors. These factors always they occur in pair. In dissimilar pair, one pair dominates the other. What is the second law state means? It is called law of purity of gametes or called as law of segregation. It is called purity of gametes. What does it state? At the time of gamete formation, gametes segregate independently. We had capital R and small r. Gamete formation is segregated as capital R and small r. Then only we perform the Punnett square. So law of segregation is called as law of purity of gametes, which states that at the time of gamete formation, gametes they segregate independently. Okay, children. So with that, I'm going to stop the class for today, children. Now what we're going to do now is, in the next class, we're going to do a test cross. We're going to see test cross and incomplete dominance in part 4 and part 5. With this part 3 slide of genetics 1, that is principles of inheritance and variation till monohybrid cross is over. Thank you very much. Please do subscribe. Thank you, children.